Hi everyone, my name's Gareth Miles. I'm a researcher at the University of St Andrews. Today I'm going to be going through a presentation that I recently delivered for a Dundee LEARN event for motor neuron disease Scotland. So I'm going to be telling you about work that my lab is currently engaged in. We're trying to better understand why motor neurons die in motor neuron disease, why they degenerate, and in understanding that better, we hope to reveal new targets for much needed new therapies to treat this devastating disease. So as the title suggests, I'm going to be telling you about some of the stem cell-based technology we use and how we use that to try to understand better what goes wrong in motor neuron disease and how we might eventually treat it. So I want to start by explaining how neurons work. So first of all, this is an image showing a motor neuron, one from a mouse spinal cord actually, that we've filled with a special fluorescent molecule so we can visualize and see all of the neuron under a microscope. So they're quite complicated structures. But this version below is a stylized version, a schematic of a motor neuron that I've drawn, just to show you some of the main parts and try to explain how the neuron does what it does. So first of all, we have the main body of the neuron here in the center, the sort of powerhouse of the neuron, if you like. And then we have this long wire-like projection that comes out of the neuron. We call this an axon. This is like an electrical cable that enables the neuron to send signals to other types of cells. That could be other neurons, or in the case of motor neurons, that would be signals being sent possibly meters away to muscles and limbs. So if it was a neuron talking to another neuron, they form this connection, which we call a synapse. So that is the connection between two neurons, or in the case of motor neurons, it could be between a motor neuron and the muscle. So the way that neurons work throughout the brain and spinal cord is they use electrical signals. We call these electrical signals action potentials. So the action potential is an electrical impulse that travels down the axon to the synapse, this connection between the neurons. Once the action potential, this electrical signal, reaches the synapse, it causes the release of things called neurotransmitters. So these are chemicals that carry the signal between the two cells. And these neurotransmitters, they get released and they bind to things on the receiving cell, in this case the receiving neuron, and they bind to these proteins we call receptors. So the neurotransmitters bind to the receptors. That then causes an electrical response in the receiving cell. And so now this can continue. And you can imagine this electrical signal can travel from neuron to neuron to neuron through complicated circuits. Again, in the case of motor neurons talking to muscle, the signal in the receiving muscle cell would cause an electrical impulse that would then make the muscles contract. So just up above, this is what these action potentials look like. You'll hear them called spikes often by neuroscientists. So these spike-like shapes represent single action potentials traveling between neurons and really conveying all the signals we need to think, to move, to walk. So that's how neurons work, but there are also other types of cells in the nervous system that contribute, and these are called glial cells. Glia basically means glue. These are the cells that we think kind of hold things all together in the nervous system, but they do a bit more than that, we've realized over recent decades. So I'm going to focus on a particular type of glial cell called an astrocyte. So these reddish colored cells here are our astrocytes surrounding the neurons, surrounding the synapse, that connection between the neurons. One of the jobs that the astrocytes do is they essentially soak up, they vacuum up the neurotransmitters after they've been released at this connection, and then they can give them back to the signaling neuron so they can be reused. So this cycling of the neurotransmitters is very important. On the one hand, it helps to halt the signal because we wouldn't want the signal to just continue forever. And it also, by recycling the neurotransmitters, allows the signal to occur again when needed. We also know, though, from even more recent work that astrocytes can release transmitters of their own. We call these gliotransmitters rather than neurotransmitters. And these transmitters, shown here, can actually directly affect neurons by binding to specialized receptors. So the astrocytes are actually having quite a key role, we think, in the function of the nervous system within lots of different, what we call neural circuits. So before going further, I just wanted to show you what it looks like in the lab, the kind of experimental setup. So this is an example of the various equipment we use in the lab, some old, some new. Looks quite complicated, but people seem to manage over time learning how to use this kit. So this is the kind of equipment that we would use. Over here you see the microscope to visualize cells and record their individual electrical activity. So before getting further into the experiments, just a little bit about motor neuron disease. So we know that motor neuron disease affects upper motor neurons in the brain, as well as lower motor neurons in the spinal cord and the brainstem. And both those types of motor neurons will degenerate, will die off slowly in the disease. And unfortunately, that leads to a loss of control of the muscle. Importantly, interneurons, which are other types of neurons that sit near the upper motor neurons and the lower motor neurons, are also affected in the disease. And as I said, the net effect of this is a loss of control of muscles. We can no longer send the electrical signals that we need to muscles to make them contract, and we have a loss of movement. So within the spinal cord, I've already mentioned that we have these motor neurons and astrocytes working together, and also we have these interneurons, other types of neurons. So together these are forming complicated networks that control our movements. So we've been trying to understand how the properties of these cells in these networks might change and how that might lead to the loss of motor neurons and the loss of function in motor neuron disease. So one of the ways we've been doing this is using specialized human stem cells. So many years ago now, Yamanaka and colleagues based in Japan discovered a way to create what we now call induced pluripotent stem cells, or iPSCs. They discovered that by forcing cells to express a certain number of genes, they could turn, in this case, a skin cell, which you could get as from a simple sample from a patient or a healthy individual. You could force the skin cell to turn back into a stem cell. So every cell in our body starts as a stem cell when we develop. So this enabled us to create stem cells that we could then utilize in the lab. Now, a stem cell has the potential to turn into any type of cell in the body. So this, of course, includes motor neurons and astrocytes. So we and others have been able to use these stem cells that have been made from skin samples from patients with MND and healthy individuals, and we've been able to take these stem cells in the lab and grow different types of cells involved in MND that we want to study. So in our case, that's been motor neurons and astrocytes. So here's an example of some motor neurons at the top, labeled with different colors for some of the different proteins they express. And here's examples of astrocytes that we've been able to grow from these human stem cells. So I'm going to describe a bit of work where we've tried to understand how astrocytes might be involved in the loss of function and the degeneration of motor neurons in motor neuron disease. So we created stem cell derived astrocytes. So we took these human stem cells, we made astrocytes from them, we grew those in the lab in our dish. And then on top of those, we took stem cell derived motor neurons and we grew those on top of the astrocytes. So now we have astrocytes and motor neurons growing together in the lab. We importantly then use a special technique called patch clamp electrophysiology to record the activity of the motor neurons that we've grown on top of the astrocytes, because we wanted to study their function. Are they behaving in the way they should? Can they produce the right electrical signals, these action potentials that I mentioned earlier? So in this study, we used astrocytes that had been grown from the stem cells of a healthy control individual. Also astrocytes grown from the stem cells that had been made from 3 different MND patients. And we had one extra group of astrocytes that had been grown from stem cells that were from a patient, but in which the gene, the mutant gene that was causing ALS, had been removed. So this was another form of control. And then on the motor neuron side, we actually just used healthy motor neurons that came from a control healthy individual stem cells. Because really what we wanted to see here was, what do astrocytes affected by MND do to otherwise healthy motor neurons? So here's some of the data that we got from this study. So on the left here, you see different types of output generated by these stem cell derived motor neurons. So the bottom is the type of output we would expect, the so-called repetitive activity. So this is a repeating spikes. So lots of these action potentials, these electrical signals that motor neurons need to produce to control muscle contraction. But some cells only showed a few action potentials, some only one, and some none at all. So even though they looked otherwise normal and healthy, they couldn't generate these spikes that they should. So we looked at the ability of motor neurons to generate these different types of output depending on the type of astrocyte that they were grown on top of. And so that's shown in this graph. Our scientists love these graphs, so I'll try and explain it. Here we have blue is the number or the percentage of motor neurons that we recorded their electrical signals, but we saw no spikes, no action potentials. The red motor neurons where we saw a single spike. The green, we saw a few spikes. And the purple, we saw what we really would expect from the most healthy, well-developed motor neurons, this repetitive firing, lots of spikes. So we have here motor neurons grown on healthy astrocytes. In the middle, motor neurons grown on MND astrocytes. And on the right, motor neurons grown on astrocytes that have been gene corrected. We'd removed the ALS mutation. And they show some differences, but they're not statistically different at this point. This is when we've grown these things together for 3 to 4 weeks. But if we grow these cells together for longer, so now 7 to 10 weeks, now we start to see some really clear and significant differences. The motor neurons that, remember, were from healthy individuals' stem cells, but grown on MND astrocytes, you see this blue bar is much larger, so many, many more of these cells are not able to generate the proper electrical signals, these action potentials. And we saw that even earlier if we looked at about 5 to 6 weeks grown together. So a bit of a complicated graph, but here I'm just highlighting the really important bit is this blue area, which denotes the percentage of cells that can't generate the right electrical signals. And you see this is much greater in the healthy motor neurons grown on MND astrocytes compared to the healthy motor neurons grown on control astrocytes and gene-corrected astrocytes. So what we're seeing here is that when we record the electrical signal of these otherwise healthy neurons, we can see proper electrical signals that would be required to make muscles contract. If we then grow them with MND-affected astrocytes, we see that these cells, these motor neurons, are less able to generate the kinds of electrical signals that they need to. And if we wait longer, this gets progressively worse. Many of them cannot produce the signals, and we think that might eventually lead to their degeneration, to their loss. But this leaves some big questions. What are the mechanisms here? What is it the astrocytes are doing to the motor neurons so that they lose their ability to generate the electrical signals that they should? So what enables a motor neuron to produce the types of electrical signals that it needs to? This is governed by special proteins called ion channels that sit on the outside of the motor neuron. And these ion channels allow the flow of what we call ions in and out of the neuron. So these could be sodium ions or potassium ions. Their flow in and out of the neuron is what creates the electrical signal, the action potential. So they're a bit like electrons flying around a wire in a computer, but in the body, it's these ions that travel in and out of the neuron. So this has to happen to generate these spikes, these action potentials. And we can record the flow of these ions in and out of the motor neuron. And that's shown on the left here. This is an example. These squiggly lines basically show you this flux of ions in and out of the neuron. And if we measure these in the presence of MND astrocytes, we find that there is some dysfunction in these ion channels in the motor neurons when affected, when grown with the MND astrocytes. And you can see that on the left here, much smaller magnitude, smaller flow of, or less flow of ions in and out of the motor neurons. So, big question again, what's going on here? Are we losing these ion channels? Are they still there? Are they dysfunctional? These are key questions that we're trying to answer. So in the last part of the talk, I'm going to describe some work we've been doing to try to correct these problems in the ion channels in motor neurons, to try to enable them to produce these electrical signals that they should again. So we've been targeting the ion channels, and we're utilizing knowledge we've recently gained which suggests that certain enzymes are important for controlling, regulating the activity of these ion channels to make sure that they are working properly. And so this is to ensure that things like the sodium can flow into the cell and create these electrical signals. So we think that loss of activity of these enzymes may be causing trouble with the ion channels, and that maybe they no longer work properly and we have this loss of electrical signal. So we've done some experiments where we've blocked these enzymes and we've looked at what that does to the activity of the motor neurons, specifically to these ion channels. And this is a complicated graph, but the key thing to know here is that this is measuring the size of these currents that flow through these ion channels. And in the light blue that I'm showing here, we have the signals in a control motor neuron situation. But if we then look at the purple or pinky purple color, we have the ion channels in the motor neuron disease situation, and we can see that they are smaller. Okay, so actually confusingly down, although it's a negative value, means a larger flow of ions. And in the dark blue that overlaps somewhat with the MND, this is where we've taken control healthy cells and we've blocked these enzymes. So it looks like blocking these enzymes causes the motor neurons and their ion channels to look similar to what we see in motor neuron disease. So we then looked at how we might correct this, and we used a viral approach. So we can use viruses to deliver genes to the motor neurons. Remember, this is grown in the dish in the lab. And then we can make the motor neuron express more of these enzymes. So we've delivered the DNA that codes for these enzymes, and now the motor neurons will have more of the enzyme present. We wanted to see if that would improve the function of these ion channels. Would we get more sodium flowing into the cell? So now on this bottom graph, this is that experiment where we've used the virus. So again, the blue is the control, healthy motor neuron situation, where we have this large downward deflection here, this large current. The pink is again the motor neuron disease situation, cells where we have the smaller current, less flow of ions through the ion channels. Then the light green, which is a little hard to see because it's overlapping with the light blue. The light green is where we've taken the motor neuron disease cells and we have added the virus to deliver more of this enzyme. And now, quite excitingly, we saw that the currents, the function of the ion channel seems to have recovered very close to what we see in the control healthy cells. So this is an exciting first step, lots more to do, but it does suggest that maybe there is some worth in pursuing this further to see if we can really recover the signaling, the electrical function of the motor neurons using this kind of approach. And we've just received funding recently to get that work going further, and we have a new PhD student who started to work on that, so we're excited to see where that goes. Okay, that's all I want to tell you about today. Just want to finish off by highlighting that this kind of work really requires a lot of people and large teams, so a big thanks to members of my lab, past and present, who have been involved in some of the work I've described, also to collaborators including Judith, who I already mentioned, and Siddharth and Chandran at Edinburgh, who we really started working on these stem cell based models quite some years ago. And importantly, this work couldn't be done without funding, and so a big thanks to various funders, including MND Scotland and the Motor Neuron Disease Association, for helping us to complete this work. So thank you very much for listening.